Right. And Yilin has been awarded many prestigious awards for his excellence in science, including there's many of the I will select some of to get your impression, some such as the Young Researcher Award from the Chinese University of Hong Kong, the RGC Research Fellowship, Research Excellence Award, and the Explorer Prize from the New Cornerstone Science Foundation. So today we are going to explore those emergent orders from the living active matter, specifically from bacteria, those beautiful patterns from the living cells. So now let's welcome Yilin to give us the talk, and the floor is yours.
All right, we should start. Thank you, Feng Tong. Yeah, it's my great pleasure to join this seminar series. I appreciate the invitation from the organizing committee, especially Feng Tong, to share our work about bacterial self-organization here. And as Feng Tong mentioned, my lab primarily uses bacteria, especially multibacteria, to trying to understand how order emerges in living systems. As many of us are aware of, living systems are characterized by the ability to self-organize. There are many examples, especially during embryogenesis, formation of somites periodically, segment by segment, and the ordered motion of cells to develop into organs, and synchronization of taste-making cells, all manifested the remarkable ability of living systems to self-organize. In addition to these examples from the so-called multicellular organisms, simpler living things like single-celled bacteria or amoeba are also able to build complex, spatial-temporally complex structures. For instance, what we are looking right now here in this video, can you imagine this is built by micron-sized rod-shaped bacteria that only knows a movement pattern that goes back and forth? It's called Mesococcus, Mesobacteria. Millions of Mesobacteria aggregate, collide with each other, and exchange signals with neighbors and build this complex pattern. So this pattern can be built in several days, and the size is on a millimeter scale. In fact, the understanding of order in living things was made primary in the framework of active matter theory in the recent 2 decades. Active matter theory has become an important framework to understand the self-organization process of living things. In general, living active matter includes any material systems that consist of units that can convert local free energy input into mechanical work. So it naturally includes all living things, but it also includes those self-driven materials or units, such as molecular motors derived from living things. And it also includes synthetic self-propelled particles. But among these, sorry, I see the message.
No, I'm sending a message to make sure people have any questions will raise their hands.
Right. I forgot to mention, if you have any questions, feel free to interrupt me. Either drop a message in the chat box or ask the host to forward the questions. So among these different systems, still bacteria has been the premier model system to understand how order emerges in active matter systems. And let me show. So let me go to the next slide here. For instance, many bacteria have a so-called flagellar motility, and as you can see, they use helical-shaped filaments appended on cell surface to drive their motion through a fluid. So they convert chemical energy from the surroundings into mechanical work. And as an individual, they consume local free energy input. And as a collective, they can make use of this local free energy input to build spatial and temporal structures. Flagellated bacteria is a very typical example of active matter. And in fact, the field of active matter was in the dawn of that active matter field was marked by this seminal paper by Visat and co-authors in 1995, where they proposed the so-called Visat model. More or less the same time, the same group of authors were studying the pattern formation of bacterial colonies, where they model individual cells, individual bacteria as random walkers. And these cells follow reaction diffusion dynamics while performing random walk. So I would say there, so this seminal paper on physical review letters must be partially inspired by their earlier work on the bacterial pattern formation in colonies. So compared to animal cells, eukaryotic cells, bacteria are very abundant. They are easy to manipulate. There are many different techniques from physics, chemistry, and biology to manipulate them. And more importantly, they are relatively simple. They are easy to model, both in physics and mathematics. So I would argue that bacterial systems or bacterial-based living matter can serve as a prototype of living matter model, just like the ideal gas model, to help us understand the physics of living systems. In addition to the relevance in physics and biology, the study of bacterial-based living matter will also help to design engineered living materials with novel mechanical functions. And here in this talk, I would like to introduce a few examples of ordering in bacterial living matter. In the first example, I would discuss a mechanism where cells couple signaling called chemotaxis and an external potential field to form spatially ordered structures. And then I will discuss a purely physical mechanism that would lead to large-scale pattern formation. And near the end of the talk, I will discuss how coupling microscopic force generating processes can lead to spatial temporal order in bacterial-based materials called active solids. So here's about the first part of the story. We know that bacteria can make use of communication, a form of communication called chemotaxis, to build beautiful structures. Here is a video taken by people in Hauerberg's lab. Here the cells perform chemotaxis towards the chemical gradients established by cells themselves. So in the end, if you are lucky, you'll be able to observe patterns or dots that arranged in space in a hexagonal pattern like this. So another so when this chemotatic bacteria is coupled to an external potential field, like here in particular the gravitational potential field, they can develop a pattern called bioconvection. So the principle behind this bioconvection process is that bacteria tend to migrate upward to see the to see the oxygen gradient, and when they accumulate near the top of a fluid film, their negative buoyancy would trigger an overturning instability, and they would sediment and drift downward. So this would generate a flow circulation, and this flow circulation can lead to the self-assembly of a pattern that looks like images shown here. So this one is the side view, and this one is the top view. The brighter parts represents regions with higher cell density. I think I got a question here. In slide 8, why are those dots bigger when they are far from the center? Let's get back to slide number 8. I think you are asking about the these dots, right? Okay. So if we look at the video, if we look at the video, initially the dots are more or less as the similar size. I think this picture was taken at the very late stage of the pattern formation process. So at that stage, these dots, these clusters were formed earlier, and they already started to run out of nutrient locally and may start to disperse. Cells belonging to those earlier dots might migrate to join dots at the outer area. So that would lead to a reduction of size. I'm not sure whether that answers your question. Okay, all right. Then let's continue. So when coupling chemotaxis and gravity, swimming bacteria can form these so-called bioconvection pattern, these variations, regular variations of cell density throughout a two-dimensional or quasi-two-dimensional fluid film. Not only multibacteria, in fact, many other types of swimming organisms, like algae, green algae, would also form this type of bioconvection pattern. And still the patterns studied in previous research, like here, what we see here, are not very regular, or although they display some characteristic wavelength. So we wonder whether we can make this pattern more regular. And my former student, Siyu, did an experiment. So let me check the question. What is the food distribution? Is there a chemical gradient towards the interface?
Yilin,
yes.
Yeah, maybe we can check all the questions later, so make your presentation can be a flow as a whole.
Oh, sure. Okay. All right.
Yeah. And later,
unless we have some.
I will check the questions for you one by one. Yeah.
Okay. If there are some very urgent questions, then
yeah.
I can answer right away. So let's address those most of the questions later. So here, my student Siyu did an experiment. He filled a PDMS chamber, a fluid chamber with a very dense suspension of multibacterial called T substance, and then enclose the chamber so that no evaporation occurs, no disturbance occurs. And what you have is that this initially homogeneous suspension spontaneously develops some inhomogeneous of cell density. And these regions, these darker spots represent areas with a higher density. And initially, these darker spots are irregularly positioned, but their position becomes more and more regular. And over time, they would interact with each other, they would fuse with each other, or they can split. So the individual dots or individual high density regions, they are like individual motile entities that interact and communicate with each other. So at this stage, the organization of these dots was more and more developed, and if we quantify the development process of these patterns, these dots, we will see that they so over time the order is so they are more and more ordered, and we can quantify this order by measuring the hexagonal order parameter defined here. And what are these dots? These dots, first of all, they are of a higher cell density, and they are in fact a column of high cell density region. So in the field of light connection, it's called plume. So if we go back to the schematic shown earlier, it's a vertical column of a higher cell density formed by the flow circulation. So here these plumes, they are consisting of motile cells, so we call them active plumes, and they form an apparent lattice pattern, so we call it an active plume lattice. In addition to measuring the pattern of these plumes, we also measure the flow field associated with these individual plumes. So we scan over the entire depth of the fluid chamber and measure the in-plane flow, the solvent flow velocity distribution, and then calculate the divergence of every plane. So we can see that across the entire column, where height 0 represents the bottom, height 500 micron represents the top. So starting from the top to the bottom, we see that the upper portion serves as a sink, okay, where fluid converges. The lower portion behave as a source of the fluid flow. So the flow is moving away from the center of the column. And by integrating the gradient of vertical component of the velocity, we can calculate the vertical component of the flow field. So the vertical flow profile looks like this. So most of the fluid acceleration, by the way, notice that this vertical velocity has a negative sign, meaning that these velocities are moving downward. But we only care about the magnitude here. So most of the acceleration of the fluid occurs at the upper portion of the column. So it manifested that the driving force for the overturning instability are primarily located at the upper portion. So that's expected for the mechanism of overturning instability. Then we looked at how the system's parameters will control this pattern, the sub-assembly of these active plume lattice. An important parameter in bioconvection is the so-called Rayleigh number, which is defined as the ratio between buoyancy-driven convection to diffusion. So this dimensionless number was borrowed from the field of the study of thermal convection, like Rayleigh-Banach convection. Earlier studies by Hill and co-authors in 1980s found and so we have an expression for this Rayleigh number in settings relevant to bioconvection. The Rayleigh number in bioconvection is proportional to the cell number density and the third order power of chamber thickness. So we can vary this cell density and this chamber thickness respectively. So we found that the cell density would not change the order itself, but it would change the spacing or the wavelength of the plume lattice. So as you increase cell density, the spacing between these dots, these columns, becomes smaller and smaller. Here, the upper panels, these are phase contrast image. The lower panels are fluorescence image. So here in fluorescence image, the brighter dots represent the columns or the plumes. So by tuning the chamber thickness H, we can systematically vary this normalized plume wavelength lambda defined here. So we found a parallel relation between lambda and this Rayleigh number. So this parallel relation was not reported before in either experiments or in numerical simulations of bioconvection. All right, so here I would like to wrap up this part of the story. So here we found that by coupling chemotaxis and an external potential field, motile bacteria can build self-assembled structures of regularly spaced active plumes. So since bioconvection occurs in natural settings, we believe this regularized large-scale flows associated with the active plume lattice may have impact on the behavior, the life of other microorganisms that coexist within the same fluid layer. So if you are interested in more details of this work, you can check our recent paper in quantitative biology. So for the next part, I would like to switch to a mechanism we recently found via which purely physical interactions can lead to large-scale order in bacterial living matter. We know that a simple spatial confinement can order active matter. For instance, if you put many rods that can grow in a one-dimensional channel, these growing rods tend to align with the boundary, with the walls, and eventually you have a very high pneumatic order along the channel. So these rods can be our bacteria, non-motile ones. If these bacteria are motile, then as shown by Ray Goldstein's lab years ago, these motile cells in circular confinement can develop spontaneously into vortices. And if they are confined in a racetrack like one quasi 1D channel, spontaneous unidirectional flow will develop. These ordered structures formed by informing bacterial suspensions are on the scale of tens of microns to roughly 100 micron. So these are mesoscale patterns. But in biology, in biological systems, we have seen many examples of very large scale pattern formation that involves the formation of regular patterns in space of a macroscopic, like centimeter scales. For instance, in the case of bacterial chemotaxis pattern that we have seen earlier, the entire pattern occurs on a standard petri dish, which is 9 centimeter across in diameter. This involves signaling between cells, right? That is chemotaxis. But are there any physical mechanisms that can lead to the formation of very large scale spatial order? There, such patterns, such order patterns were predicted for active Brownian particles that undergo the so-called motility-induced phase separation. But in reality, in experiment, patterns formed attributed to motility-induced phase separation were more or less like what we see here. So we can hardly tell whether these patterns are regular, although we do see some signature of phase separation, but it appears that they are randomly distributed in space. Could that be more ordered? Recently, my former postdoc, Hao Ran, who's now an assistant professor at Zhejiang University, found a physical mechanism called self-enhanced mobility that can lead to centimeter scale ordered pattern in bacterial films. Here he looked at quasi two-dimensional dense bacterial film supported by a solid substrate by agar. So this film spontaneously developed a lattice of vortices, and these vortices are arranged in space in a highly regular manner. So here, what we are looking at is a video, as shown by the timestamp here. As you can see, from the the velocity field or vorticity field, the patterns, the vortices are highly stable in space. So each vortex has a size roughly 300 micron, consisting of about 10,000 cells. They are highly stable, and they do not need any spatial guidance or spatial confinement. And they form spontaneously. So this lattice displays hexagonal order as can be quantified by calculating the pair correlation function or the triplet distribution function. As I mentioned, these vortices, the formation of these order arrangement of vortices does not require any spatial confinement or spatial guidance. And individual cells within the vortices are free to cross the boundaries between vortices. If we check the movement of individual cells, we can see that they enter and leave the vortices quite frequently, but they display different motion pattern inside the vortices, the vortex phase, and outside the vortex phase. When we looked at the development of this large scale, centimeter scale vortex lattice, we found that it emerges from a very disordered active turbulence-like structure. So initially, we see many transient vortices emerging in the field of view, and over time, these transient randomly located vortices would move and interact with each other and coarsen, and eventually, these vortices are stabilized. So during this process, the correlation, the lifetime of these vortices grew longer and longer, starting from just a few seconds, like in the typical active turbulence of bacterial suspensions, to up to several hundred seconds. And during this stabilization process, the cells is gradually separated into two phases. One, the vortex phase, the other, the disordered phase outside the vortices. And in these regions, they display distinct speed. So in the vortex phase, their speed is much higher compared to the disordered phase. So what's the origin of this? We found that there's a strong correlation between individual cell speed and the local polar order of their neighbors' movement. In fact, this correlation is so clear that it can be approximated as a linear correlation. So if we measure single cell speed and its local polarity order, polar order, it can be well fitted into a line. So this implies that the mobility of individual cells, which is defined here, mu, so basically it's the coefficient between the relation of in the relation between single cell speed and the self-propulsive force. So the single cell mobility is proportional to this local polar order. So this implies the mobility of individual cells depends on the arrangement of local neighbors' movement. If the local neighbors are moving in an ordered manner, they appear to have a higher mobility. So we could understand it as, we could interpret it as the fact that individual cells' mobility is enhanced by the collectively generated active stress due to the movement of local neighbors. Since the polarity order or polar order is a proxy of local collective active stress. So we put forth this idea into a wizard type particle-based model. So in this model, we modify the speed of particles by incorporating this relation between mobility and local polar order of the neighboring particles. Here in the model, we also include equations for the orientation and the angular velocity coupling. So simulations with this model produces two phases. One, the disordered turbulence-like phase. Two, the lattice phase, the phase that associate with the formation of large-scale and stable vortex pattern. Here is an example of such a simulation. So starting from randomized configuration, this model gradually evolves a lattice of stable vortices. And this pattern, the formation of the development of this vortex space critically depends on the coefficient beta here. We call the mobility enhancement coefficient. So for a given activity or mean self-propulsive force, there is a threshold coefficient below which you only see turbulence-like behavior. And above this coefficient, this critical value, you see the ordered lattice space. You might argue that the vortex lattice we obtain here in this particle-based model is still not very similar to the experimental one. It's less ordered compared to the experimental one. So we attributed this fact to the missing of hydrodynamic interaction between particles in this model. If we incorporate that using a continuum description of the active fluid, we can produce highly regular hexagonal pattern of vortices. So if you are interested in that part, you can refer to our paper. But the main message is that here we suggest a purely physical mechanism that can lead to large-scale vortex pattern in living systems. Such large-scale vortex pattern has been observed along neural tubes during embryo development. It's called somatomeres. So these are basically vortices of cells in the developing tissue in an embryo. It may also be related to the vortex-like pollinase movement during the primitive tree formation. So we hope developmental biologists would be interested in this idea and may come up with ways to verify this hypothesis. The reason of this idea is that these systems are also consisting of high densely packed individual cells, and there the cell's mobility will be closely related to the local the self-generated active stress. All right, still have some time. I would like to switch gear and talk about the spatial temporal ordering in bacterial-based active solid. So if we embed motile bacteria in an elastic matrix, that would make a system solid-like, in which all the individual particles, individual motile cells have relatively constant separation, but collectively they drive the local mass element into motion. And if we embed flagellated bacteria in an elastic matrix, we could realize interesting forms of global motion patterns, such as oscillatory translation or oscillatory rotation. So these would serve as continuum materials for understanding the large-scale emergent mechanics of active solids. But here I would like to focus on another way of coupling microscopic force generating process. So in this process, individual cells do not move by rotating flagella. Instead, they move by grappling hook-like motile organelle called pullus, type 4 pulla. Pullus is a linear filament extending from a cell's surface. As shown in this video, they extend and retract. So pullus can generate forces up to 100 piconewton. It's the strongest molecular motor known today. Instead of looking at the motion produced by these pileated cells, we looked at how this large-scale force, this pulling force generated by pilus motors, can produce emergent mechanical behavior in a dense solid-like bacterial film. So my former PhD student Siqi found that in such a bacterial film driven by pilus motility, one can observe a striking spiral wave pattern, as shown in this video. Here we see that steady spiral waves propagate from the center of the film. So notice that the scale bar is 500 micron here. So the entire system is several millimeter in size. The period of the spiral wave is usually several minutes. What's the nature of this spiral wave? Is it any chemical wave or other forms of wave? Before that, let's look at how this wave develops. So starting from initially featureless state, we see that some localized oscillation appear, and these localized oscillation would propagate in space, forming segments of wave fronts. And these wave fronts gradually merge and annihilate with each other, eventually leaving a few highly stable spiral waves. So this can also be found by looking at the space-time evolution of the spiral center's trajectory. So this vertical axis is the time. If we make a slice at a specific time, the positions of spiral cores are located on this two-dimensional slice. And as time evolves, only several spiral cores will remain, and their trajectory now appear as straight lines, meaning that they are highly stable starting from these times. So this spiral wave is formed by a self-organized process. And to further understand the nature of this wave, we looked at the microscopic picture during wave propagation. I'm not sure if you can see it clearly from the other side, but here I'm showing the cell density variation during the propagation of the wave, and also the motion of mass element during the wave propagation. So the wave propagates from lower right to the upper left, and the mass elements move back and forth during the propagation. So this is a longitudinal wave associated with density variation. If we treat this bacterial film as an elastic film, we can show that the phase variation, the cell density variation would be related to the displacement, and will be related to the rate of displacement. So if we plot the displacement rate in green and the cell density variation in orange, we can see that they are perfectly aligned. So this tells us the wave is basically a tension wave that represents the spatial temporal regulation of tension across the entire bacterial film. So to understand how this wave emerges, we resort to the well-known core-motor model that describes the synchronization of phase oscillators. In the classical core-motor model, we have a set of phase oscillators, back whose phase are coupled in a sinusoidal manner. But here in our case, a classical core-motor model of this type can only produce global synchronization. That would not be spiral wave. So to account for the spiral wave, we note that individual microscopically, the Pulis motor are highly irreversible. It can be, it means that it can accelerate in phase, but it will not retract in phase. So we introduce an important modification to the core motor model. We made it non-reciprocal. So we added a step function like term, a coefficient into the coupling function here. The step function depends on the sign of this coupling term. If it's negative, we take a value 1 minus epsilon over 2. If it's positive, we take 1 plus epsilon over 2. So this epsilon belongs to, it ranges from 0 to 1. It's a measure of the level of non-reciprocity. So in the case of epsilon equals to 1, then that's full non-reciprocity. In that case, the oscillators leading in phase would affect the oscillators lagging in phase, but those lagging in phase would not affect the ones leading in phase at all. Okay, so with this change in the core motor model, we performed simulations and found that the emergence of spiral wave critically depends on the existence of this non-reciprocity. If epsilon is 0, we would not see any spiral wave, but if epsilon is a finite non-zero value, we can see spiral waves that are highly stable, just like what we see in this experiment. Now we notice that the value of epsilon also controls the separation or the final stabilized number density of the spiral cores. Why is that? We found that there is an intermediate range repulsion between the spiral, as represented by these blue triangles. So two spiral waves, if they are very close, then they would attract and annihilate. But if two spiral waves starting from different locations move towards each other at the intermediate separation, they will feel repulsion, and this repulsion guarantees that they will not get too close. So as you increase the level of non-reciprocity, the range of with intermediate range repulsion, okay, this range expands. So that explains why for higher level of non-reciprocity, we have a so the spiral cores, the spiral waves can be more stable, and we have a higher number density of spiral cores. All right, what's the implication of this spiral wave? In many systems, including chemical, non-equilibrium chemical reactions and biological systems, we can observe spiral waves, as shown in a range of in an array of images here. The unique feature of the bacterial spiral wave we found here is that it's a mechanical spiral wave. It's a tension wave, spiral tension wave, and its core, the motion of the core is distinct from those known in in non-equilibrium chemical reactions, like BZ reactions. Here, the spiral cores are almost stationary. It does not display any meandering motion, like those familiar in BZ reactions. Instead, it shares some similarity with the with certain types of spiral waves found in the cytoplasm and in the electrochemical waves in cardiac tissue. So we suggest that this type of mechanical spiral wave may provide mechanical analog to understand the origin and the control of stable spiral waves in other more complex systems, such as in heart tissue. All right, to summarize here, we have introduced several physical mechanisms that can lead to spatial and temporal order in bacterial-based living matter. In particular, we mentioned mechanisms that can lead to very large-scale order in bacterial-based systems. In general, the formation of large-scale spatial temporal order structures will require complex biochemical regulations or genetic regulations, such as by the mechanisms listed here. But here, in the few examples I described here, purely physical interactions can do the job, and we hope this can help us better understand the biological pattern formation across different length scales. And more importantly, we hope this work will help establish bacterial-based living matter as the prototype model to understand the physics of living things, especially for processes related to non-equilibrium phase transition. Finally, I would like to thank my lab members whose work has been mentioned in this talk, including Shiqi, Haoran, and I forgot to put Siyu's name here. And also would like to thank my collaborators over the years, whose help has helped us to get to here. And also want to thank the funding opportunities from the Hong Kong Special Authority Region Government and the National Science Foundation of China, as well as New Cornerstone Science Foundation. If you are interested in any of the videos I showed in the talk, please feel free to check out our lab website. We are hosting most of the videos on our website. All right, I would like to thank you for your attention and be happy to take any questions. So maybe I can check the chat box.
Yeah, Professor, thank you, Professor Wu, giving us really exciting lectures and telling us those emergent orders from those bacteria. And it's exciting we can regulate these orders using mathematical models and equations. So now let's try to keep the questions. Remember this the master from first panel, the question from Maharana. She's asking, what is the food distribution and is there a chemical gradient towards the interface?
Hi, Maharana. Yeah, good to see you here.
Hi, very interesting talk.
Thank you. So the question was about
I think it was slide 10, yes, around the slide 10 or 9, the chemotaxis part. I was wondering if the competition was within the layer, or was there a gradient outside towards the interface?
So here the cells are seeking oxygen gradient. So oxygen gradient is pointing upward. So the top is higher in oxygen concentration, and the bottom
So
the bottom of the column is depleted with oxygen.
I see. So the oxygen is diffusing into the layer and then it's
Right,
consumed by that. Thank you.
right.
We have another question from Ron Maria. Do you unmute yourself? I saw you raise your hand.
Yeah, hi. I don't know if you can hear me.
Yep.
I'm on my phone. Okay, good. Thank
Hi.
you for the hi. Thanks for the great presentation. I actually had the similar question, so you answered this one already. My other question is what were the bacterial species that you use throughout your different experiments? Oh, I just found how to switch on my video. Sorry. So yeah, what were the species you used, and are they do you know why they have this bioconvective behavior? They obviously they need to get oxygen. You're saying that there's a gradient of oxygen, so they want to go at the top to get the oxygen. But the fact that they drag the oxygen down with them as they sink down through bioconvection or with gravity. I don't know if it's gravity or if it's just swimming towards the lower oxygen concentration. Do you know why they do that in connection with their metabolism?
Yeah. So so speaking of why they do that here, this so the bacterial species we use here is called B subtilus, and B subtilus is quite strictly anaerobic, meaning that they like to stay in an environment ample in oxygen. So here, if oxygen is of a higher concentration at the top and lower concentration here, then their intracellular signaling pathways would lead them to move up towards the higher concentration region.
Oh, sorry, I misunderstood what you said. They are anaerobic. They are strictly anaerobic bacteria, right?
Sorry. So they are aerobic.
Oh, aerobic.
So,
okay.
Yeah.
So they want to go up where the oxygen is. So I understand why they would go up.
Yes.
Are you saying that the bioconvection is just a byproduct, like is not something that benefits their metabolism? Because why don't they just stay at the surface then? You know what I mean? I'm trying to understand the relevance of this behavior for the metabolism or the ecology of these species. I'm trying to understand if it's basically something that has evolved to improve their fitness, to improve their access to their nutrients, or if it's just an emergent property that may not benefit them at all. And it's because of the experimental design that they display this bioconvective behavior. I don't know if my question makes sense. I'm not a physicist, I'm a biologist, and I'm recently got interested into
that's a very good point.
into
bioconvection. That's a very good question. So so talking about the ecological relevance, here naively one would think that if they accumulate near the top and then passively due to gravity's pull passively sediment down and drift down, this would seem to reduce their ability to acquire more oxygen. But think the other way, if cells are if the cells are during the process can generate flows, the flow circulates and pull and make the oxygen distribution more homogeneous across the entire fluid film or the entire fluid depth, then it will benefit the entire population. Yeah,
that makes sense.
Would that make sense?
I guess it would. Yeah. No, absolutely. Thanks.
Yeah. And by the way, I saw some recent work that studied the ecological relevance of this bioconvection for bacteria. So they put different types of bacteria and see how they are arranged, and they found that different types bacteria with different I forgot the exact nature. Bacteria with different properties tend to accumulate in different regions created by this bioconvection pattern, and that has a real ecological relevance.
Yeah,
Mm.
I'm sure it has potentially a lot of relevance in nature, and I would love to understand that a bit better from our perspective. That's what we are trying to do. So thanks for the great presentation and for explaining it, everything. That was very, very helpful. Thanks.
Thank you.